Okay, I think we'll go ahead and get going. So good afternoon, everybody. My name is Barb Lee from the National Farm Medicine Center, and it's my privilege to introduce our speaker today for our scientific seminar. This is being videotaped on MediaSite, and we know there's some people that are attending a different ORCA Lunch and Learn, so we expect that they'll be watching this presentation a little bit later or right now from their office. So. I've known Matt Nonnenmann for more than a decade, and he's been a friend of ours here at the Farm Center for a long time. He's well connected with 3 of the other 10 regional NIOSH egg centers, specifically the centers in Colorado and Texas and Iowa. And Matt received his master's and his PhD from the University of Iowa in industrial hygiene. And he currently is an assistant professor at University of Iowa with the expectation to be promoted quite soon to associate professor. And there at University of Iowa, he teaches several courses in industrial hygiene and mentors graduate students, as well as running his own research program. He's got a lot of experience in agriculture, and it's quite impressive that he knows a lot about the dairy industry, swine or pig industry, and poultry industry as well. And his research aims to help us understand what's in the air that the animals and humans breathe, and then how to manage adverse aerosol exposures in livestock production buildings, mostly large animal confinement buildings. And in the future, we expect and hope that Matt will be advising us here at the Farm Center as we build an agricultural microbiome repository for future research endeavors. So please join me in welcoming Matt Nonnenmann to our podium.
Thank you. Thank you. Is this working okay? You guys hear me? Okay, I think I can hear myself, so I had to double check on that. So yeah, anybody who can spell my name 3 times quickly gets a prize at the end of the presentation. So it's quite a long name. I usually get lots of comments, and when I call or I usually just start spelling it immediately because people usually can't spell it. So, but I'm originally from the Quad Cities, Rock Island, Illinois, Moline, Davenport, home of John Deere Tractor. Anybody know about that? A few of you are nodding. Yeah, that's where I'm from. So I grew up there, and my grandparents farmed. My dad was a dentist, but he had a hobby farm, and so I was around agriculture a lot, but also around science, and I was interested in human health, and so it kind of morphed. Instead of going to Iowa to go to go to dental school, I went to Iowa and learned about, wow, corn dust causes lung inflammation? I had no idea. I thought it was just asbestos that causes lung problems, and so that's kind of what started my interest. And then I learned about industrial hygiene, which is what this is a certified industrial hygienist, for those of you who may or may not know that, but it's a professional credential, kind of like a professional engineer or CPA, and so you're certified in skills to measure human exposure. And so often, rather than saying I'm an industrial hygienist, I say I'm a human exposure scientist. So try and measure what people are exposed to. And often I've traditionally focused on inhalation exposures in agricultural work environments. And so today I wanted to talk to you about bioaerosols. Basically, it's a type of aerosol that has biological components in it, rather than just say silica, which is sand or dust from sand. You guys in Wisconsin, have you guys have that talking about silica at all recently? Yeah, I think I've heard a little bit about that with the frac sand mining and that sort of thing. So community exposures. But today we're going to look at the organic components of aerosols. I kind of created an outline to kind of take us through the talk. It's, our talk is, a good 50 minutes here, so I wanted to help keep us oriented. So I'm going to introduce you to what is a bioaerosol and how do we characterize a bioaerosol. Is it, target specific? Do we look at everything in the aerosol? And then some past or ongoing research projects, related to poultry production or chicken production. Some target-specific projects where we tried to measure virus in swine production. And then the third project, which is a switch from agriculture to healthcare. You guys, this is a hospital-type environment. I figured you'd be interested in that. Is there any physicians in the room? No physicians in the room. No free lunch for the... Okay, we've got one. Good. You're going to represent all physicians for us today, okay? And then we have related to toilet flushing and patient care, and some of the aerosols that are generated during toilet flushing. And then the last project is a project that's currently ongoing and funded through the CDC to try and control bioaerosols in swine production. Okay, so first topic, what is a bioaerosol? So basically it's an aerosol that contains abiotic or biotic agents. Abiotic is like a chemical that's produced, or a protein that's produced by the bacteria in the aerosol. Some examples are endotoxin from gram-negative bacteria, or muramic acid is something that's also measured in samples as an indicator for gram-positive bacteria, or the biotic component, which is the actual organism itself, so like a virus or a specific bacteria. And there's renewed interest in these types of exposures given the microbiome work that's happening, the lung microbiome, the gut microbiome, and that sort of thing. And actually that's kind of what I've done, is I've taken some of the techniques that have been used for those sorts of studies and tried to apply them on the exposure side. So often this field is called aerobiology. If you Google it, you can learn more about bioaerosols. Okay, some settings where some bioaerosols are present. I mean, this is not an encompassing list, but clearly agricultural is one, healthcare is another, other sorts of processing type industries where you have organic materials like wastewater, compost, food processing where there could be microbiological growth, like we're making yogurt, or the dairy industry, et cetera, baking. But for us, this is kind of two primary examples we're going to focus on. One is a bioaerosol that's generated from a person, like during a sneeze. And then, anybody have a guess what's on the right up here? Right? Chickens, right? So chicken production. Who likes chicken? I love chicken. I have no self-control when it comes to fried chicken. Is anybody else here like that? I just eat it like crazy. So I really like chicken. And this is chicken production. We have to make a lot of chickens to satisfy our desire to eat chicken. And so this is how they do it. And there's about 30,000 birds in this one building. And it kind of a little bit of description of the building. You can see the buildings are typically dark to keep the bird activity down. And then you can see the ventilation down here. It's tunnel ventilated, so you have these big fans on the end, and the inlets come in on the side. And then the birds are often separated by some barriers, and as they get bigger, those barriers are removed, and they can move throughout the building as they wish. And then, important point here. So this floating stuff, this is in Texas, so this isn't snow, this is dust, right? So it's really dusty, and there's some gases in that environment as well. Okay, so whenever we talk about exposure, we're really interested in aerosol size, because size indicates where the penetration will, or where the particles could potentially deposit in the lung. So as we look at different examples of bioaerosols, your pollen here, this is the category of aerosol, and this is the size of the particles. So 100 microns here is kind of the benchmark. That's where stuff starts to get into your upper nose airway and lungs. So, bacteria kind of span that boundary, and cat allergens, viruses, we're way down here, really small stuff. So understanding the particle size of what your target is for the size of the aerosol is really important from an exposure standpoint. And we have samplers that represent, that will collect everything below this line. And so those are important to use those when you're interested in exposure, because we want to know what gets inside the person. And the other really important point is when you have really small particles, it takes them a long time to settle out of the air. You have dead air, no air movement, kind of like in here, right? Someone sneezes, you generate some really small particles. This virus kind of floats around for a while. So that could be that could impact exposure, for people. Or if you're just generating large particles, those settle out fairly quickly. So that's important. And this last point down here, diffusion time. So basically that's movement due to Brownian motion, which is the movement of air molecules. So you have like really small particles, they can be pushed around by the air molecules even. So, so that's the phenomenon we're describing there. Okay, so that's kind of what a bioaerosol is. We're going to talk a little bit about characterization. So, quote, total bioaerosol, which is a term that's used in the literature, or total bacteria measurements, and then target-specific measurements. So often when people measure bioaerosol, they'll look for total bacteria. And this benchmark has been used in the literature, and it's often reported in colony-forming units per cubic meter of air. And there's some recommendations that, if the concentration is above 1,000 colony-forming units per cubic meter of air, that's bad. No one really knows who came or why that number was established, but that's something that's reported in the literature. And then when you look at OSHA, who kind of regulates work environments, there's no recommendations for exposure. It's kind of a problem. If you know anything about bacteria, some bacteria, we eat them because we need them for our gut. And other bacteria, we don't want to eat them, so a little bit is bad. So it's difficult to have exposure benchmarks that are just based on the number of colonies. And typically that's often used for a general growth media, like trypic soy agar. And you can also do sampling with more specific agars, like McConkey agar for gram-negative bacteria. So I'll show you an example of this. So when we're interested in doing bioaerosol sampling, this is a very commonly used sampler. It's an impaction-based sampler, so you put an agar plate in the sampler with whatever target media you're going to use. You turn on a pump, it's hooked up to tubing, you pull air in, the air goes in here, goes through these little holes, and then impacts on the agar. And this is the flow rate, 28.3 liters per minute, and then you take that plate and you stick it in an incubator. And then you count the colonies after a certain amount of time, and you calculate. Remember, our number that we were looking at was colony-forming units per cubic meter of air. So you do the math. There's 1,000 liters per cubic meter. You can do a calculation for concentration. This is kind of the classic way that bioaerosols have been measured in the past. Here it is. We're doing it in the chicken barn. So stationary sampler. And here's some results from the chicken barn. There's a lot of bacteria in the chicken barn. I mean, literally, I would turn on the sampler for 30 seconds. I know we have at least one microbiologist in the room. Can you count all those colonies on there for me? Yeah. Okay, good. Well, it's kind of challenging, right? There's a lot of stuff in the air. Yeah. So, as we decrease our sampling time, you can see that it's like we're still right there, right? These plates are loaded. And this is just, this is a general growth media. And then this is McConkey agar down here. Again, we're seeing fewer gram negatives, so yeah.
Question.
Yeah.
Those plates look like very pure colonies. They're all the same type.
Yeah. Sure. It's a great question.
You expect to have different colonies.
Right. So I think, I'll talk about this in another slide, but, there's obviously some, if you are in an environment where the conditions, there's multiple bacteria, and one type just grows a little faster than the other. I mean, this incubation period was really short. So if that bacteria grows faster than the others, it's going to just outcompete, and you're going to get no signal from the other bacteria, and the plates can be overloaded. So that's my explanation. So in other words, and I'll talk about this on this slide, right? Some methodology gaps. So you have a culture bias, so it limits the characterization. Basically, you can count what grows under the conditions used in the experiment. You can't count what doesn't grow. And then organisms grow at different rates. So you throw it in the incubator for 12 hours. Oh my gosh, the plate's all white. It's like, oh, well, were there other bacteria in there that are not this type that were outcompeted virtually immediately in this sample? And so this culture bias is pervasive in the literature. And then also you, of course, only count what remains alive after the sampling, because sampling stress may destroy some bacteria. So there's error in our measurement. That's the bottom line. There's just a lot of error. And when we're trying to look for exposure benchmarks, you don't want a lot of error in your exposure measurement. You want to be really specific in what you can measure so you can look for health associations with certain exposures. That's how public health science works. You know, you measure an exposure, and you look for frequency of disease in the people who are exposed, frequency of the disease who are in the unexposed, and hopefully you see some sort of dose response. And then you go, okay, 1,000 colony-forming units, that's the bad number. We get above that, we start seeing problems. We haven't been able to do that in this field. So the other thing, the other concept is, you know, a lot of the work has been based on stationary sampling. So have you ever been to a farm? Does a farmer stand in one spot all day? No way, right? None of us do. So kind of a industrial hygiene approach is using personal sampling. So you have a pump on the person with a sampler, and they go and do their thing. And then you have a sample that represents where they were, what environment they were in that day. And then hopefully you use a sampler that has size selective characteristics. So you're also measuring what goes into the lung. Okay, so I was thinking about this, obviously, with projects. And so now we're going to talk about projects. So this is a project that's completed. It's in the chicken production. Here's the list of the co-authors on this particular project. Kate O'Brien is a postdoc of mine, and then a bunch of other folks. Then I'm at the last. I'm on the last there. But you know, some of the other folks are from Texas A&M, Mississippi State, and Stephen F. Austin State University, who did make the NCAA tournament this year, which is great. They're doing a good job. And then some folks from Iowa. Okay, so if you know anything about broiler production, most of it doesn't occur in the Midwest. There is a little part up here in Wisconsin. Anybody know what they're doing up there? Maybe turkey, yeah. So yeah, I noticed that on the map, and I'm like, oh, I guess there is some in Wisconsin. But you know, most of it is in the southeast down here. And so when I was in Texas, I thought, okay, well, what type of agriculture do they do around here? They do broiler chicken production. Okay, let's go do something in broiler chicken production. And you know, there's a lot of farms, about 250,000 workers. And kind of the goal of the study was to determine personal exposure to inhalable dust and endotoxin during work tasks. So that was one goal. The secondary goal was to characterize microbial composition of those samples. And so we're going to talk more about the second goal today, because you know we only have a short amount of time, but we did do both. And this is kind of the experimental design. So we had, again, our personal exposure sample to inhalable dust. We had 69 samples, and they were all from different farms, which is a big deal. It's hard to get on farms these days. And we used an inhalable sampler. So it has a 50% cut point at 100 microns. Remember that 100 micron benchmark we were interested in? And you know, the flow rate, the type of filter. It was task-based. We were looking at, they were doing some litter sampling on the farm, and then they were doing another job called mortality collection, where literally you walk through all the chickens every day and you pick up the dead ones, you haul them to the end and throw them out of the building. And when you have like 30,000 chickens, you get a few that die every day. And so you have to go in and haul them out. It's a daily task. And then the other thing that we did is that we looked at settled dust in the barn. And again, the microbial composition. This wasn't an air sample. This was something off the surface. Do we have any toxicologists in here? No? Well, in the field of toxicology, often if you're looking for the impact of a certain substance on a tissue culture, you'll often collect settled dust from the work environment. And so settled dust may look a little different from a microbiological point of view. And so I thought we need to include settled dust in here, because that's what a lot of the inflammatory, or the scientists who explore inflammatory processes, looking at lung tissue, that's what they'll use, a settled dust sample. And so this was our sample analysis process. Have our dust samples, then we went through, we weighed them, we analyzed some for endotoxin using a recombinant factor C assay, and then we did DNA extraction, purification, quantification, library preparation, then ultimately whole genome sequencing on the Illumina HiSeq for this, and the University of Minnesota did that for us, because we didn't have this capability at Iowa. And this is the results. So in the literature, there's kind of this blanket statement of what's in agricultural dust. Usually people say, oh, it's skin, it's corn, it's it's bedding, it's, it's like literally they copy one sentence from one source and put it in the next publication. You see the thing over and over and over again. And I'm like, all right, we're going to look at the DNA in the sample. And we're going to tell people what's in the sample instead of what's been used in the past is like a microscope analysis, just looking at particles. So maybe you guys can help me out here. My Latin's a little bad, but so that one's pretty easy. So most of the DNA on the y-axis you can see was microbial, right? So there's a lot of microbes in this dust. And then Gallus. Chicken, right? Chicken, good. Zay, maize. Corn, good. Homo sapiens. People, right? Yeah, so there's some people DNA, and the rest of it is from our sequencing process. And then you can see the three types of samples here. Black is litter sampling, gray is the settled dust, and white is the mortality collection. So, there's a little difference. Obviously when you're picking up lots of dead birds, there's a little bit more chicken DNA in the air. That makes sense, I guess. So, fairly similar. And then when you start to break it down by domain level taxonomic information, eukaryotes, so it's like people, chickens, archaea. So was anybody in high school or took micro when this category was established? You have to be younger than me because they didn't talk about this when I took micro. So this used to be a type of bacteria. Now it's its own thing. Correct me if I'm wrong. That's my understanding. You're nodding. Is that right? Did I get it right? Okay, good. All right. And then viruses. So we have like about 25% of the genetic material were from viruses, and the remainder was from bacteria. So bacteria are clearly dominating this aerosol. And here's some, our top 25 from this aerosol. And this is relative abundance. So your scale is red is high, and then black is low, and this is the top 25. And across the top here, you have our colors, our litter sampling colors, so that's black. These are the samples from litter sampling. These are the samples from mortality collection, and these are the samples from the settled dust. You can see that, clearly there's different profiles, based on settled dust. I mean, settled dust doesn't quite look the same as these other personal aerosol samples. And some, there's some bacteria that kind of jump out that are really common. Staphylococcus species AL1, for those of you who know what that is. We're not sure what the impact of that is yet. Lactobacillus crispus, some Salmococcus, a few more, Staphylococcus lentis, Lactobacillus, and Enterococcus cocorum. So those were kind of the most common among the samples. So it's, in other words, does this give us more information than the total bacteria measurement? Heck yeah, this is gobs of information. So much we don't really know what to do with it yet. So, we can go through and pick out these individual organisms and see, oh, well, is this specific organism associated with, a pathogen, or is there some opportunistic pathogens in here? One thing that was interesting to me was right here, Pseudomonas phage. Lactobacillus phage. Anybody know what a phage is? It's a virus, right? So what sort of virus, what's its host? Bacteria. So it's like, so in our bioaerosol, we're being exposed to viruses that infect bacteria. It's like, and it's not like a little exposure, it's like in the top 25, right? So it's like, phages, there they are again. What does that mean? I don't know. But I think it's interesting. Okay, and so when, my colleagues did this principal component analysis, found similarities between the mortality collection samples and the litter sampling, and the litter sampling samples, the air samples, and the settled dust was kind of all over the place. So in other words, they're pretty different than everything else. And so we kind of use this and argue in the paper that when you go to these environments and you collect dust off of settled surfaces and you apply it to a cell culture and look for inflammatory responses, you kind of don't know what you're going to get, and it's pretty different from what people are breathing. So that may not be the best approach. Maybe we should sample the air and then apply what's collected in the sample to the tissue culture. Okay, so some conclusions. Again, dust is primarily composed of microbes. There's statistical differences in microbial populations between aerosolized and settled dust, and then lower variance in microbial populations among the air samples, which kind of makes sense. I mean, to me, if you have a settled dust sample, that's being heated and cooled, and they're out competing each other, the bacteria are developing their own, kind of who's boss in that own little settled community. So the profile of the bacteria is going to change where it's static, where it's not moving. Okay, this work's been published, and it's online, open access, so you can go and look it up if you want. And then, definitely thank you to all my colleagues, my postdoc and the Southwest Center for Agricultural Health for the funding. Okay, so that's that study. Now we're going to talk about something more specific. So then this was general characterization. We're trying to figure out what people are breathing. And now we're like, okay, let's switch a little bit and let's look for something specific, like influenza virus in swine production. Okay, so again, this is something that's published. My postdoc and I did this work. We did, again, personal samples on people working in agricultural environments where we thought that there was a specific target we were looking for, like influenza. Anybody get a flu shot this year? Right, so there's a pretty hardcore public health effort to prevent the transmission of influenza. I mean, we've all heard about it. Most of us probably decide to go do it. My wife's a nurse, and she doesn't like to do it. She has her own opinions about that. We argue about that all the time. So we won't talk about vaccines right now, but I, of course, am a huge proponent of vaccines. But, okay, well, that's not the purpose of this talk. But anyway, this, in my opinion, was a route into the human population from pigs. And so I started asking the question, well, where are pigs produced? Obviously, they're in Iowa. Iowa's number one in the nation, and then North Carolina, a little bit in southern Minnesota. This is the inside of a finishing barn, kind of the last stage before they go to market. And for those of you who aren't familiar with influenza, influenza is a zoonotic illness, it's a virus that infects people and also animals. And this is a great figure, influenza. Thought it was great. I copied it off the internet. Here's the address. If you want to go use it. But it's really neat. And here's the interaction. This is people, and this is pigs. And then, we've got our H3, which is over here, and then our H1s. And where's HN2? So, there it is right there. Okay, so we've got this overlap, people, pigs. And there's like six pigs for every person in Iowa. And so it's a lot of pigs. It's a lot of virus. And so how is it transmitted? Direct contact. So we started talking to vets and say, well, when pigs are sick, how do you go in? They go into the building and they take an oral fluid sample and send it off to the vet diagnostic lab. Then that sample gets tested. And they say, yep, they got influenza. Or if there's like a surface, that somebody touches that's contaminated, then they, of course, obviously touch themselves. Or if there's an aerosol, and there's some evidence of virus in the aerosol, in an aerosol environment in pig production. So our question was, all right, where's the virus and who's being exposed? So obviously it's, the vet's going to treat these animals. And, is it inhalation, direct contact? So we started digging into this, and we're like, okay, what are the methods that are used to measure inhalation exposure to virus? And so this is a setup where the person wears a sampler and they wear a pump. And this was an experimental sampler developed by NIOSH, and how it works is that the air supply comes from here. This goes to the pump. The air goes in this little hole, and it swirls around inside this centrifuge tube. And then it jumps over here, swirls inside this centrifuge tube, then it goes back through here where there's a filter. And so they wear this on their person. And then there's another sampler that's strictly filter-based. It's just like it looks. The air goes in that big hole and it impacts on the filter. And so we're like, okay, well, instead of using other things, can we use this to measure exposure? And what I like to do on studies is I like to use more than one sampler so that you can compare, how they work. And so one, you get characterization and quantification, and two, you can compare across samplers and seeing which one works a little bit better. So basically, we had the vets go in and do some nasal swabbing of pigs that were already diagnosed with influenza. So we had a few vets that we called, and they'd call us. They're like, hey, I got a farm that's hot. And we're like, okay, we'll meet you there. So we drive there, and we'd meet them. We'd hook the stuff up on them. They would go into the farm. They would do swabs for about 30 minutes, and we would hang the samplers on them. And then when they came out, we would analyze the sample and look for virus. And I'm just going to explain this table a little bit to you here. So we did this on 5 farms. It was a small sample size. And then this is that NIOSH 3-stage sampler I described to you. So each stage has a certain size of particle that it'll collect. And so like the 15 ml tube, for those of you familiar working in a lab, the kind of bigger tube was the 15 ml tube. And this is the virus that we found. So on a farm where it was like the day of diagnosis, it was hot, virus was hot that day. In the 30-minute sample, there was like almost 5,500 copies in that first stage of virus. This is based on qPCR.
500 or 5,000?
I'm sorry, 5,000. 5,500. About 5,500 copies right here on that first day. And then, the middle stage, where there's a range of like 4 microns to 1 micron in size, there was fewer, but we were still detecting it. And then, down here on the filter, the filter didn't do so well. We struggled with detection on the filter. And so when you combine all of these stages together, that's what you get in this column over here. And again, on that first day, second day, you can kind of see how the concentration goes down as the disease progresses and they get better. But you can see how the concentration decreases based off of, time elapsed after initial evaluation. So there's virus there. When they go in to collect these oral fluid samples, you've got like a bunch of pigs in that building, 1,500 pigs, and they're all sick. A good chunk of them are sick. They're coughing, hacking. They're generating aerosol, and people are breathing it. And so then, that opens up other questions. And then, of course, our filter-based sampler, we didn't do quite as well with that sampler. It didn't work as well. So, what this study told us is that, 1, we found virus, so virus is in an aerosol in the building. And then 2, if we're going to sample for it, we should probably use the NIOSH sampler when we do that. Okay, so I kind of jumped ahead a little bit here. The NIOSH sampler performed better. The 15 ml tube accounted for most of the virus. And then, the virus is really small. And so we think it was bound to other particles, other larger particles that would impact out in those larger stages. And that's what we were seeing. So otherwise, everything would have been on the last stage on the filter that collected the smallest stuff. Or maybe filter's horrible and you can't get any virus off, and it was loaded with virus and we just couldn't find it. That's another possibility. Okay, and so this is somewhat questionable, but I found some infectious dose information for influenza in the literature. And so the literature claimed 100 to 650 RNA copies. Again, it's like a PCR-based level, which, it's like, how do you argue that that's an infectious dose? But anyway, if that's true, if it takes 650 influenza copies of RNA to infect someone, the workers would receive an infectious dose fairly quickly in the building based on the concentrations we measured. Limitations, few measurements, only a few farms, and swine vets were reluctant to participate because they make a business out of meeting the needs of their clients, which are the farmers, and they don't want to cause problems. So they were pretty reluctant. We had 2 vets that worked with us closely that were old enough they didn't care. So it was tough to do this. Okay, so next step, again, we're going to keep working on our methods to improve our methods, further characterize influenza aerosol, which is really, really neat. We've been trying to do it in the hospital. Haven't had a whole lot of luck there. But when you're in a building that has a ton of animals that are sick, it's easier to find. And then lastly down here, controlling the exposure, engineering controls. That's what kind of led us to the last project I'm going to talk about. Okay, so now we're moving to healthcare. So we've done ag, general characterization, ag specific. Now we're going to do healthcare, general characterization. And we're going to focus on toilets. You know, there's no toilet lids in the hospital, none. So the question was, are we getting aerosols off these toilets? And the answer is yes, it is. So I jumped ahead, I told you. But here's the team. So the UI Epicenter personnel, some infectious disease docs and staff who worked on the study, nurses. Sam Knowlton, she's a student of mine who's graduated. She spent a lot of time in the hospital waiting for poop in the toilet and toilets to be flushed. So she deserves all the credit. A little bit of background. Healthcare-acquired infections cost a lot of money to a lot of people. They're very, very expensive. And hospitals are motivated to prevent these because Medicare does not cover the cost of treating a healthcare-acquired infection. And so the hospitals have to pay for that, and they don't want to pay for it. And so prevention, this is where the prevention model really can, people encourage us to come and learn more to try and prevent these infections. Obviously, they want to prevent them because people get sick and die from them as well. And so people have GI illness, they often go to the bathroom more. Anybody ever had a GI illness? Or you're like running to the toilet all the time? It's like, oh my gosh, you know, that means you're flushing more and, you know, lots of variables. So our study objectives, you have to bear with me here a little bit. We had 3 conditions. We had no waste, no flush. So in other words, we go into the patient room, there's no waste, no flush, we're just going to sample and get background data. We had a second condition. There's no waste in the toilet, but we're going to flush it. We're going to see what's in the water that's in the toilet, which is also pretty gross, actually. And then loose fecal waste with flush. So that's the third category when there's waste in the toilet, and we're going to go in there and we're going to flush it. And so we have 3 categories, and we measured just basic particles over time and distance from the toilet, particle concentration, and then we looked at total bacteria. And our goal was to see, are we measuring a bacterial plume? So again, a total bacteria measurement, which doesn't tell us a whole lot, but it tells us, okay, we're generating an aerosol that contains bacteria. Okay, so these are the categories of the particle size bin. So we measured really, really small particles, 0.3, 0.5, 1 microns, 3 microns, and the largest we measured was 10 microns. And we summed the number of particles per minute. So we considered the flush as like the point source. So we measured before the flush, during the flush, after the flush, and then we sampled for 5 minutes. We switched out the plate, put in a new agar plate, another 10 minutes, switched out the plate, put in a new plate, and then sampled for 15 minutes. So we had a total of 30 minutes of sampling around this one flush. And then we incubated all the samples, and we did this at 3 distances from the toilet, 0.15, 0.5, and 1 meter. And those are just kind of arbitrary distances that we chose. And we were curious, was there a difference across time? Was there a difference across distance? And then was there a difference across category, like no flush, no waste with flush, and waste and flush? And then so we did, again, a total bacteria measurement. And then the next thing that we did is we started targeting people who had C. diff. Anybody know what C. diff is? Yeah. So it's a big, big problem. It's a healthcare-acquired infection. Often it's antibiotic resistant, and it kills people. Makes them really sick in the hospital. And so, and it's pretty resistant in the environment. And we're trying to find out how is this bacteria moving around the hospital. We think toilets may be contributing to that. So we're, again, going to the hospital, not only trying to find people who have GI illness, but we're trying to find people who have C. diff and waiting around their room for when they go to the bathroom, and then we move our equipment in. So that takes even more time. But we do have some data. Okay, so our sampling cart, it's like this scary thing right here. It's basically 3 cases with 3 pumps in it. And here's our samplers. I showed you pictures of those earlier, how they work. We have media inside the samplers, and they're on this little shelf, and it's mobile. So we got to just wheel it in there, plug it in, set it up, turn it on. We built all this in-house in the lab. And then here's just some example of some plates. Okay, so how it worked is we would turn on the particle counter right here, the OPC, and then we would flush the toilet. There you go, there's the toilet flush. And then we would, of course, sample the whole time. Everyone awake now? Do you have that toilet flush? And then, the researcher was right here. And these bathrooms are often pretty small. So, and here's just a photo of what it looked like when it was happening. And if you want to go to YouTube, you can find that on YouTube. Okay, so the data that for under the condition where there was no waste and no flush, there was no change in particle concentration across time. So I took that slide out so you didn't have to look at that. But this slide is there's no waste, no poop in the toilet, and it's with a flush. And so on the y-axis over here, we have average particle concentration. And on the x-axis, we have time in minutes. And then here, this dotted line is when we flush the toilet. And then the red line is when we change the plate in the sampler, plates in the samplers. So you can kind of see here, there's a little bit of a peak after we flushed. Minute 4 was the flush, right? Or minute 3, I'm sorry. And so minute 4 was kind of the peak. And then things would kind of normalize after that. Then here's fecal waste with flush. And interestingly, we saw a little bit more variance in particle size in this data set, and we think the consistency of the poop in the toilet impacted how much aerosol there was. If it was a really, really loose stool, we saw more aerosol. If it wasn't, if it was like thicker or whatever, then we saw less. But that was just kind of anecdotal observation. I'm sorry I even have to say that, but it was based on the graduate students' notes. And so we think that may explain some of this. But we did see increase in particle concentration. And again, it was like these smaller sized particles that really changed over time. And then when we did an analysis where we looked at, we compared particle concentrations over time. Remember, our baseline before we flushed was minute 1, 2, and 3. So we compared concentrations in minute 1, 2, and 3 to minute 4. And we found these statistical differences here across particle size. So I threw this up here just to show this to you. It's pretty kind of busy. There's lots of p-values. But we did compare across minutes. And, there was no change in particle concentration here. But this is the kind of the money table, right? So when we compared the bioaerosol concentration, we measured about 210 colony-forming units per cubic meter. And the no waste, no flush, that was just background, just no one being in there. And then, which was different, statistically different from when you had fecal waste with flush. And when we compared across time and across distance, we found no difference, but just across category. So no waste, no flush was different from fecal waste with flush in concentration. Again, just total bacteria. So we're generating some sort of bacterial aerosol from the toilet. And that's not a new finding, but what's new is like this was in a patient setting with ongoing patient care, and no one had done that before. And so then these data haven't been published yet. We're still collecting these, but so when we started looking at C. diff, so there was nothing in the toilet. We just went to a room with someone who had C. diff. We pushed in the cart, we flushed the toilet, and we sampled. This is what we found so far. Again, these numbers are low, but in 2 of the 10 trials, we detected C. diff. That's the concentration. And then when there was waste in the toilet, 1 of 3 trials, we did find C. diff as well. And these numbers aren't quite, I think we're around 17, maybe 18 now for our sample size. And I know we've detected C. diff at least a couple more times, so I didn't have those updated numbers for this slide. So we're finding it. That's the important thing. And then the question is, well, is it the same bacteria from the patient that's in the toilet? Is it the same bacteria that you find in the toilet, like on the counter or at the nurse's station or on the floor? We have to do sequencing to kind of get a feel for that. Okay, so again, limitations. Few cases of C. diff, selective selective agar, culturing conditions. There's additional variables, to consider, like how long the toilet was idle, And generalizability is a challenge because we just did this in one hospital. And we don't know how it would compare to other hospitals that may have different toilets. But surprisingly, the toilets are pretty similar across state hospitals. You know, they must have used the same vendor. Okay, kind of conclusions. Analyses suggest that flushing increases small particle concentrations, and differences in conditions suggest loose fecal waste, previously used toilets, aerosolize organisms, and we didn't find any difference across time or distance. And then, of course, we're detecting C. diff. Okay, you made it to the last one. Bioaerosol exposure control. This one's the shortest because this is an ongoing project. It's an engineering control. It's a ventilation project in swine production. And I can't share with you the technology we're using because we're applied for a patent and they tell me I can't say anything to anybody. So I just have a blue box that describes that. But the goal is to design and test a ventilation system to reduce dust and microbes in swine production. Remember how we measured influenza in there? And we're like, oh, this may be a problem. Well, what do we do if we have a room full of pigs breathing virus? How do we control that? How do we keep it from moving to another building? Right now, they just exhaust the air. And so how do we control that? Also, there's other viruses like PEDV, the site that we were just working with. They lost, I can't remember if it's 6,000 or 7,000 pigs, you know, and the last week we were there from this virus, and it just killed them. You know, it's like, this is a huge deal for them. And so we're hoping they let us come back because, you know, movement from and to farms transmits that virus. It's not a zoonotic virus, but we're part of that movement, and I'm hoping they still let us come back. So our goal is to improve animal health and to improve human health, kind of the One Health model. All right, so where we are in the project, we did some baseline monitoring, not 2 months ago, but like 14 months ago, and we said, is there dust there? Yes. Are there microbes there? Yes. So we answered that question. Yes, this swine building is representative of what we typically find in swine buildings. And then we built a prototype ventilation system and treatment system. And then year 2, we tested it to see, does it, I can tell you this, that part of the technology is a filtration technology. We wanted to test it and see if our flow rates were enough to reduce dust. And so we did a treatment versus control room experiment. And we did data collection. We had to wait until it was really cold. So we started on December 26th, which is horrible. And we had to go 4 days a week through middle of February. And the farm owners, because they do not want a lot of people coming to their farm, they said that they only wanted me to come to their farm and study staff, like 2 other study staff, and that was it. So I had to go on every single field visit, which is painful for a PI, right? And we're not supposed to do that. We're supposed to be in our offices working, right? But I had to go on every single field visit, early morning, shower in, shower out, and make this thing happen. So how it worked is we had 3 sampling locations, kind of stationary sampling locations, just to measure ambient concentrations in the room. And we pull air from the room into our bioaerosol control device, which is outside the building. It's mobile, it's in a trailer, it's deployable. And then we treat it and we blow it back into the building through this sock. And this is a farrowing building. So we're trying to make the room air better for the piggies in there. And these are the mamas, and they have little babies in each crate. And so here's where the air is sucked outside and treated. This is where it's blown back in. And it's completely contained. And so the goal is to be able to respond quickly and deploy this device. And you can see you can run it right through the window right there. So you don't have to cut a hole in the wall, which farmers really like when you don't have to cut a hole in the wall of their building. Okay, so summary and conclusions. So bioaerosol exposures occur. We're trying to figure out the impact of that on human health. And then there's the tools to measure them need a lot more work. We need to use molecular biology to help us characterize these aerosols and examine the relationships between exposure and impacts of human health. And so it's really underexplored, and I have really enjoyed doing this work. So I guess I can open it up to questions. We have a few minutes left for the audience, and thank you for everyone. Yeah.
One theory out there that influenza is so much worse in the winter is that public buildings have very little humidification. And so when they're aerosolized, they stay in the air longer. What about, was there any consideration here of the effect of relative humidity in these buildings and in your work?
Right, so it's a great point. And relative humidity is argued about in the literature quite a bit. And so I like to think of it as so our spit is a little viscous. Sorry if I get too detailed. Our spit is a little viscous, and when you sneeze in a really humid environment, those particles will increase in size because they absorb water. So, and then they'll fall out of the air quicker. This is my opinion. This isn't proven. I call it like the Goldilocks scenario. You know, like Earth is in the perfect Goldilocks from the sun, right? The perfect area. It's not too hot, not too cold. Same thing with humidity. When you sneeze, there's a certain, I think, what happens is if the air is too dry, it'll completely dry out that saliva and it'll deactivate a lot of the virus. But I think that there's a range of humidity where the particles will shrink a little. They'll form a little bit of drier protein around, and then in the middle of that particle, you have this perfect ampule of virus. You know, like the gel caps you can get for Advil? Kind of like a similar concept, you know? This is my opinion. I haven't designed an experiment to try and prove this. Just thinking about it a lot, that's what I think is happening. So then when the conditions are really perfect, you know, and people sneeze, it like decreases the particle size, they can penetrate deeper into your lung, and then it's like this perfect little protected ampule of virus that deposits, and then it humidifies in your lung, increases in size, and then delivers the virus. That's what I think happens. But I haven't, maybe someone here can help me design an experiment to test that. But that's what I think happens. And so there's a lot of argument about relative humidity. I think if the air is really dry, I think, you know, it'll help you. But if it's that perfect, you know, 40%, kind of a guess, 40% relative humidity where it dries a little, but doesn't completely dry out the particles right away, I think that's what happens.
You showed a chicken house there with fans on one end and ventilation openings on the side. What's the difference between the air next to the fans and the air way the hell at the other end of the place?
Well, the air near the end of the fans is really bad, really bad. The concentration of dust, because it's a linear system, so the air moves in one direction, right? So as the dust is generated on the floor of the building, it all kind of concentrates near the fans. So if you're standing down near the fans, you're getting loaded with dust compared to if you're right up at the front of the building.
So if you had a building where you pulled it in, say, multiple intakes from the roof, and then shot it out the side, so you don't have, you know, 300 feet airflow.
Yeah, I mean, I think that'd be great. You know, the producers don't, when they design the buildings, they're thinking completely about animal production, and so they don't quite consider human exposure. And I was, you know, working with folks who set standards for those buildings, and they're not really interested, you know, in changing a whole lot, just for the person.
Well, most infectious disease has a lot to do with inoculum size. I think the animal health would be impacted by how much junk is in the air.
Oh, I think it definitely is. And I know if the fans stop working, all the animals will die. You know, it gets too hot and they have no fresh air and they will die. So they have to move enough fresh air through there to keep the animals alive. And they have alarms on the fan, so if like the fan goes off, they get a phone call, you know.
So down south, do they have to do any air conditioning in the summertime?
So they use evaporative cooling pads on the sides of the building, and so they'll pull air in, you know, through these evaporative coolers and to try and humidify it and cool it, and that's how they do it. So it's still pretty warm in those buildings, but it's not as warm as it is outside.
But when you use evaporative cooling, you're really humidifying the air.
Right. You are humidifying the air. Yeah. Mm-hmm. In Texas, it's not as much of an issue because the air is pretty dry, you know, and so, and I guess, well, I take that back. It depends where you are. It's pretty humid in Houston. Yeah. Any other questions?
Any questions?
With the static settled dust collection, what was the kind of protocol for where that collection device went?
So we didn't actually use a collection device. We just went to horizontal surfaces in the building and collected settled dust. And we just collected large amounts of it, and then we kind of mix it and then collect the sample for
Like
extraction.
near a human station or a human entrance, just anywhere? Or
Anywhere.
Okay, well, thank you, Matt, for this presentation. I'd like to